Now today we discuss the biotechnology principle and process. In principle and process of biotechnology, two LAQs are important. First LAQ, tools of biotechnology, and second LAQ is steps involved in the recombinant DNA technology. First we discuss the what are tools of biotechnology. Principle and process of biotechnology. Principle and process of biotech. In here, first question is tools of biotechnology. Tools of biotechnology. Tools of biotechnology. Okay. Which tools are used in the biotechnology? Okay. Then first tool is enzymes. First tool is enzymes. Okay. In here, how many types of enzymes are involved? First one is restriction enzyme. Restriction enzyme. Second one is ligase enzyme. Second one is ligase enzyme. And third one is DNA polymerase enzyme. DNA polymerase enzyme. Okay. Then first one is enzyme. Second one is what? Second tool. Second tool is agarose gel electrophoresis. Agarose gel electrophoresis. Agarose gel electrophoresis. And third one is amplification of gene interest by using of PCR. Amplification of desired DNA. Amplification of desired DNA. Okay, by using of PCR. P stands for polymerase, C for chain, or for reaction. Okay, then make a competent to the host cell. Make a competent to the host cell. Make a competent to the host cell. To host cell. And then last one, vector we discussed. Vector. Okay. Now we discuss one by one first. Restriction enzyme. Restriction enzyme. Okay. Now this restriction enzyme is discovered in where? In a E. coli cell. These are what? E. coli cell. E. coli is nothing but Escherichia coli bacteria. Okay, in the E. coli bacteria have what? Bacteriophages. These are what? Bacteriophages. Okay. What is this? Bacteriophages. Okay. These are bacteriophages. Okay. Now these are E. coli bacteria. Okay. E. coli bacteria. Now this bacteriophage growth is controlled by the E. coli enzymes. Okay. Point 1. Bacteriophage growth is controlled by the what? E. coli enzymes. Point 1. Bacteriophage growth is controlled. Bacteriophage growth is controlled by E. coli enzymes. E. coli enzymes. So how many enzymes are control the bacteriophage growth? 2 enzymes are control the bacteriophage growth. Okay, 2 enzymes are control the bacteriophage growth. Okay, first enzyme is what? Enzyme name methyl group. CH3 is methyl. Methyl group add to the DNA is one enzyme. And second one is what? Cuts the DNA. Cuts the DNA. Second enzyme is what? Cuts the DNA. Later that cuts the DNA enzyme name gives what? Restriction enzyme. Later given the name that cuts the DNA name gives the what? Restriction enzyme. Restriction enzyme. Okay. So how many restriction enzymes are discovered? How many? restriction enzymes are discovered in how many strains of bacteria? 900 restriction enzymes are discovered. 900 restriction 900 restriction enzymes are discovered. 900 restriction enzymes are discovered in 230 strains of 230 strains of bacteria. 230 strains of bacteria. Okay. So generally restriction enzymes are two types. In this restriction enzymes are two types. Don't confuse. Discovered in how many enzymes are there? How many enzymes are there? 900. In how many strains it discovered? 230. In 230 strains, 900 enzymes are discovered. But types of restriction enzymes are two types. Types are two types. Exonuclease restriction, exonuclease restriction, exonuclease enzyme, restriction exonuclease, and restriction endonuclease. Restriction endonuclease. Restriction exonuclease and restriction endonuclease. Now this restriction exonuclease and endonuclease both are belongs to the which class? Both are belongs to what a nuclease class. Nuclease class, important point. Okay. Then now we discuss the what a restriction exonuclease and endonuclease. First we discuss the what a endonuclease, later we discuss the exonuclease. First restriction endonuclease enzyme. Restriction endonuclease enzyme. Restriction endonuclease enzyme. Okay. First point. What is the function of this restriction endonuclease enzyme? Restriction endonuclease enzyme first identify the length of DNA. Okay. For example, these are the DNA. This DNA length is identify the water restriction endonuclease enzyme. For example, these are 2 meters or 1.5 meters or 1 meter or half meter. Okay, how much their length is discovered, identified by the restriction endonuclease enzyme. Point 1 is identify or inspecting the length of DNA. Restriction endonuclease enzyme function is inspecting the or identified the length of DNA. Identify the length of DNA. Okay, identify the length of DNA. And second one, after identified, then it's identified or recognize the its recognition sequence. Its recognition sequence is identified. Then identified, after identify the length, then identify the recognition sequence. Recognition sequence. Okay, identify the recognition sequence. Now what is the recognition sequence? Recognition sequence nothing but palindromic sequence. Okay, in the bracket recognition sequence, these are also important for 2 marks. Recognition sequence nothing but what? Palindromic sequence. Okay, in the bracket recognition sequence nothing but palindromic sequence, important for 2 marks also. Okay, bracket close. What is a palindromic sequence? Example, Malayalam. Example is what? Malayalam. Palindromic example is Malayalam. See, when we read the forward direction and when we read the backward direction, same letters are repeated. See, I read first forward, ma, M-A, ma. Then here also M-A, ma. Okay, then L-A, la. Okay, here also L-A, la. Okay, then Y-A, here also Y-A. Y-A forward direction, and backward direction also YA. Okay. Then LAM, LAM. Here also LAM, LAM. Okay. So when we read forward direction, backward direction, same letters are repeated. It's called what? Palindromic sequence. When we read, when we read forward, when we read forward direction and backward direction, forward direction and backward direction, same letters, same letters are repeated, same letters are repeated, it's called what? A palindromic sequence. It's called palindromic sequence. Okay. Now this palindromic sequence applied to the what is biotechnology in the DNA means this palindromic sequence have what are 6 nitrogen bases. Continuation here. Important for 2 marks. Palindromic sequence. Okay. It has how many base pairs? 6 base pairs. It has 6 base pairs. It has 6 base pairs. For example, one strand begins with the 5 to 3 prime direction, and next strand begin with the 5 to 3 direction, 5 prime to 3 prime direction. Next strand also begins with the 3 to 5 prime direction. Both strand have what are 6 bases. Both strand have what 6 bases, complementary bases. See here. Like these are what 5 to 3 prime direction, and these are what 3 to 5 prime direction. So here I have what how many base pairs are 6 base pair, like GAA TTC. Okay, GAA TTC. How many base pair? How many bases? 1, 2, 3, 4, 5, 6. Okay, here in 3 to 5 prime direction also GAA TTC. Okay, here also how many bases? 1, 2, 3, 4, 5, 6. Okay, so when we read the forward direction, like say here GAA backward direction, TTC forward direction, TTC backward direction. So palindromic sequence means it has 6 base pairs. When we read the forward or backward direction, same letters are repeated. It's called what? Palindromic sequence. Right. So restriction endonuclease enzyme first identify the length of DNA, then recognize sequence. That recognize sequence nothing but palindromic sequence. After identify the palindromic sequence, it's bind at the recognition sequence. For example, these are the DNA. In the DNA, in this region have what? In this region have what like recognition sequence are there. In this region also recognition sequence. In this region also recognition sequence. So after identify the recognized sequence, then it's bind at the where? At the recognized sequence. Now restriction endonuclease enzyme bind at these regions. Recognized sequence region, which enzyme is bind? Restriction endonuclease enzyme is bind. Then after binding, it cuts the water. Both stand in between the GA. In between the GA. For example, it's cut in between GA. Here also in between the GA. Means it break the water, sugar and phosphate bonds. Sugar and phosphate bond. Because of what is the backbone of the DNA? Sugar and phosphate. Sugar and phosphate. That's why it's cut the water. Which bonds are? Sugar and phosphate bonds are breakdown. Okay. So after cutting in between the GA and GA, what happened? Only one strand is overhanging. For example, here G is there, remaining A, T, T, C is removed. Okay. Then in here only G is removed and A, T, T, C is there. Okay. So this strand is overhanging strand. Now these are 5 prime to 3 prime direction, and these are 3 prime to 5 prime direction. So one strand is overhanging strand. This strand is called what a sticky ends. This strand facilitate the what a sticky ends means what is the sticky ends important? It joined with what other DNA. It facilitate with what other DNA. Okay, these are what it's called. What is the this totally information about a restriction endonuclease enzyme. Okay, once again revise this. First, identify the length of DNA, then identify the recognized sequence. After recognized sequence is identified, it bind with the recognized sequence. After binding, it cuts the water, both stand in between the GA. After cutting, one strand is overhanging strand. That strand is called what a sticky ends. The sticky ends provide or facilitate the water joined to the water other DNA because of hydrogen bonds are there. That's why. Okay. The next is what restriction exonuclease enzyme. Restriction exo. A name only here. Exo means outside or edges. Outside or edges. Okay. Restriction exonuclease enzyme. Restriction exonuclease. Restriction exonuclease enzyme. So this restriction exonuclease enzyme also cut the DNA at the both stand at the center region. At the center, so not center region, at the edges of DNA. At the edges, like these are also cut the what polytomic sequence, but not particular direction, not particular in between the AG. Just cut the water at the edges. Okay. So here GAA, then TTC. Here also GAA, TTC. So it cut the water edges like this. Like it cut the water these edges and these edges. Okay. GACD and like this. It cut the edges, does not provide the water, does not provide the sticky ends. Does not provide the water, sticky ends. Exonuclease cuts the exonuclease cuts the DNA, cuts the DNA at the edges, of both stand, and this exonuclease does not provide the sticky ends. It does not provide the sticky ends. Okay, these are what exonuclease enzyme. So one enzyme is over. The next is what polymerase enzyme. What is the function of this polymerase enzyme? Sorry, before that, first we discuss the only enzymes. Then now, after cutting the DNA by restriction endo and exonuclease enzyme, the next enzyme is what polymerase, DNA polymerase, also called TAC polymerase, also called TAQ, TAC DNA polymerase, TAC DNA polymerase. Okay, TAC means is obtained from the bacteria name TAC, Thermus, T for thermus, A for AQ, aquaticus, Thermus aquaticus. Okay. DNA polymerase enzyme function is polymerize a complementary DNA. Polymerize a complementary DNA. See here, in this has three steps. Okay, so indirectly it's called amplification of DNA, amplification of desired DNA by using of PCR. Indirectly amplification of desired DNA, amplification of desired DNA by using of which technique? PCR, polymerase chain reaction. Okay, in here I have three steps. In here has three steps. First step is denaturation. Second step is annealing. Second step is what? Annealing. Third step is what? Extension. Third step is extension. Okay. So before discussing these three steps, this enzyme tag DNA polymerase enzyme is obtained from the which bacteria? Okay, that we discussed. Okay. Tag DNA polymerase. Tag TAQ. Tag DNA polymerase. Tag DNA polymerase is obtained from Thermus aquaticus bacteria. Is obtained from Thermus aquaticus. Thermus aquaticus bacteria. This Thermus aquaticus bacteria, okay, is obtained the which enzyme? Is obtained the which enzyme? Tag DNA polymerase. What the special of this tag DNA polymerase? This enzyme means which enzyme? Tag DNA polymerase enzyme. This enzyme can stable in high temperature. Can stable in high temperature. Clear? Okay, now come this point. So in this process, amplification of desired DNA by using a PCR process has three steps. Step one is denaturation. Step one is denaturation. So what is the denaturation? Say, now these are desired DNA fragment. What is the concept of this? What is the concept of this? We want the what? Multiple copies of desired DNA. We have what? Initially we have what? Only one fragment, desired DNA fragment. Only one desired DNA fragment. From this, only from this one DNA desired DNA fragment, we want the what? Multiple copies. Okay, of desired DNA fragment. Okay. See, in first step is denaturation. First step is denaturation. Step one. In here, step one is denaturation. Okay. So these are 5' to 3' and these are 3' to 5' and here nitrogen bases are there. Nitrogen bases. Okay. Like A, T, G, C, A, T, G, C, A, T. Okay. A pair with the T, T pair with the A, G pair with the C, C pair with the G, A pair with the T, T pair with the A, G pair with the C, C pair with the G, A pair with the T, T pair with the A. Okay. In between AT has double hydrogen bonds. AT or TA has two hydrogen bonds, and GC or CG has what? 3 hydrogen bonds. AT has how many hydrogen bonds? 2. Okay. Or TA also has 2 hydrogen bonds, right? So here GC 3 hydrogen bonds, again CG 3 hydrogen bonds, 2 hydrogen bonds, 2, then 3 hydrogen bonds, 3 hydrogen bonds, 2 hydrogen bonds, 2 hydrogen bonds. Okay. Now, when this DNA, D means what? Loss. D means what? Loss or removed. Naturation means their natural property. Their natural property loss in this step. In this step, what happened? This DNA fragment is treated with a high temperature. In denaturation, what happened? Denaturation. In denaturation, DNA fragment, which fragment? Desired DNA fragment. Desired DNA fragment treated with a high temperature. Treated with high temperature. What happened? If these are treated with a high temperature, in between the two strands, these hydrogen bonds will be breakdown. These hydrogen bonds will be breakdown and two DNA strands will be separate. Okay. When desired DNA fragment treated with a high temperature, what happened? In between nitrogen bases, hydrogen bonds will be breakdown. In between nitrogen base pairs. In between nitrogen base pairs, hydrogen bonds will be breakdown. Hydrogen bonds will be breakdown. And two DNA strands will be separate. Two DNA strands will separate. These are about denaturation. Okay. So these two DNA strands will be separate now. After like this here. Okay, these two DNA strands will be separate. Okay, 5 to 3, then 3 to 5. 5 to 3, then 3 to 5. Okay, like A, T, G, C. Okay, A, T, G, C. Okay, let T, A, C, G, A complementary T, T complementary A, G complementary C, C complementary G. Okay. So these hydrogen bonds will be breakdown in the first step. So in the second step, what happened? Annealing. Second step, what happened? Second step is which one? Annealing. Annealing means a set of primer. A set of primer. How many sets of primer are important? Two sets of primers. Two sets of, sorry, note down here. Second one is annealing. Second one is annealing now. In annealing, what happened? Two sets of primer add to the water 3' region of both stand. Okay. Annealing means two sets of primer. Two sets of primer. Two sets of primer add to 3' region. 3' region of both stands. 3' region of both stands are called annealing. Are called annealing. See, in the primer have what has a oligonucleotides, means nothing but nitrogen base are present in the primer. Primer has what? Oligonucleotides. Oligonucleotides. Now, in this stand, which one is 3' region? This one. In this stand, which one is 3' region? This one. Okay. At this region, set of primers. How many? These are one set of primer, and these are what? Next set of primers. Okay. So two sets of primer add to the water 3' region is called what? Annealing. Okay. Now, at the primer has what? Oligonucleotides. Okay. And the DNA tag DNA polymerase. Tag DNA polymerase extend the primer. Tag DNA polymerase extend the primer. Okay. In the primer has oligonucleotides. Okay. Now this primer provides the water. Primer provides the water. Here C is there now. So primer provides the G. Then that polymerase enzyme extend to the primer from this position to this position. And here G is there. It's provide the C. Here T is there. That's why provide the complementary base A, and A is there. That's why provide the complementary base T, and here C is there. Provide the G. G is there. Provide the C. T is there. Provide the A. A is provide there. A is there. Now that's why provide the T. Okay. So these are what called extension. This step is called what? Extension from this direction to this direction. Okay. So these are 3' now. So this is 5' direction and with the 3' direction. Okay. So here also primer is there. Here also primer is there at a 3'. So like here G is present. So primer provide the C. Okay, then the DNA polymerase extend to the primer from this position to this position. Okay, here A is there. It's provide the complementary nitrogen base T. Okay, here C is there. It's provide the complementary nitrogen base G. Here G is there. Provide the C, and T is there. Provide the what? A. Okay, A is there. Provide the T. C is there. Provide the G. G is there. Provide the C. Okay, and hydrogen bonds will be formed. Double bond, triple bond, triple bond, double bond, double bond, triple bond, and a triple bond. When here also triple bond, double bond here, triple bond, triple bond, double bond, triple bond, and a triple bond. These are one DNA strand. So in 30 cycle of this PCR, form what a billions copies of desired DNA fragments. Okay, so these are denaturation, and annealing is what just add the primer at the 3' end region. It's called what annealing, and these are what extension. Okay, in 30 cycle, billions of desired DNA fragments. Desired DNA fragments are formed. Okay, these are about amplification of desired DNA or gene interest by the what? Which process? PCR, polymerase chain reaction process. Okay, Then next one is what? Gel electrophoresis. Next tool is gel electrophoresis. Gel electrophoresis. What is the concept of the gel electrophoresis? After fragmentation of DNA by using a restriction enzyme, for example, these are the DNA. This DNA is cut and forming what? Fragments, right? In this fragment, we want the water desired DNA. Okay. So what is the gel electrophoresis concept? Check or select the desired DNA fragment or check the digestion of desired DNA fragment. Okay. So in gel electrophoresis, take a water one plate. Take a water one plate. This plate is called agarose plate. This plate is called what? Agarose plate because of on the plate has a medium or matrix. That matrix is what? Agarose matrix. This matrix are what? Agarose matrix. Agarose matrix. You know agarose or agar. Agarose or agar is obtained from which algae? Red algae. Obtained from red algae. Their name is Giladium and Gracilaria. Okay. Then now on the plate has what again? Wells. These are wells. These are what? Wells. Okay. So wells are present in the plate, agarose plate, and then matrix also there in the agarose plate. Okay. Then now these DNA fragments are filled in the where? In the wells. Now these are DNA fragments. These are DNA fragments filled in the where? In the wells. So DNA is a which in charge generally? Negative in charge. DNA is a negative in charge. When apply the what? Electricity. When apply the what? Electricity to this agarose plate, what happened? These DNA fragments are moved based on their size. Okay. These DNA fragments are moved based on their size. For example, these are the DNA fragment large in size. These are moved very slow. For example, these are what desired DNA fragment are small in size compared to this one. These are fastly move compared to this one. If this desired this DNA is what again very small or lightweight, these are move very fast and reach to what plus region, anode region. Because of DNA is a negative in charge. So here charge is negative charge. Opposite to the here, positive charge means anode region. These are cathode region. Anode region, cathode region. Okay. When we apply the electricity, DNA fragments are moved based on their size. So which size DNA fragments are moved very fast and which size DNA fragments are moved very slow? Okay, large size DNA fragments are moved very fast and large size DNA fragments are moved very slow and small size or weight DNA fragments are moved very fast and reach the what anode region. Okay, now first one, take a what agarose plate on agarose plate. First one on the well or in the well, fill the water. In the well, fill the water. DNA fragments. Okay, then apply the water electricity. Then apply the water electricity. Then apply the electricity to agarose plate. The next one is what? After applying the electricity, DNA fragments are moved based on their size. After apply the electricity, DNA fragments are moved DNA fragments are moved based on their size, based on their size. Okay, large size DNA fragment, large size DNA fragments, large size DNA fragments are move very slow, or move slowly, and a small DNA size fragments, small DNA fragments, small size DNA fragments, small size DNA fragments are move very fast, and reach very fast and reach to which region? Anode region, plus region, anode region. Okay, fast and reach to what? Reach to anode region, Okay, then now this design means it's called now design. Now this fragment cannot see our eye. That's why first the desired DNA fragments are stained with the ethidium bromide. These are stained. Okay, these are stained with the what? Ethidium bromide, and then immediately expose the what? UV radiation. Okay, first is stained to the what? The DNA, and then immediately expose the UV radiation. After expose the UV radiation, these DNA fragments are appear which in color? Orange color bands. Orange color bands. Okay, then after appearing the orange color bands on the agarose plate, then these orange color bands will be separate from the agarose plate. That is called what? Illusion. It's called what? A illusion. So here continuation, DNA fragments are moved based on their size. Okay, and large size DNA fragments are moved slowly, and small size DNA fragments are moved very fast. Okay, and reach to the water, reach to the what? Reach to the anode, reach to the water? Anode region, reach to the anode region, and then after that what happened? The DNA is stained. The DNA cannot visible with our eye at the anode region. That's why DNA is stained. DNA is stained with the ET. ET was the ethidium. Br bromide. Stained with the ethidium bromide. After stain, okay. After stain, what happened? Immediately expose the UV radiation. Immediately expose the what? UV radiation. Immediately expose the UV radiation. Immediately expose the UV radiation. Then this DNA fragments. Then DNA fragment appear. DNA fragment appear which color band? Orange color band. Orange color band. Orange color band. Okay. Now this orange color band is separate. Now orange color band. Orange color band is separate. Orange color band is separate from agarose plate. Separate from agarose plate is called illusion. Is called what? Illusion. Important point. Illusion. Okay. Here illusion is important. Then ethidium bromide is important. Okay. Ethidium bromide is important. And that color is also important. Appear which in color. Okay. These are about gel electrophoresis. Then vector. The next point is vector. Okay, what is the vector? What is the vector and what is the importance of vector? And this vector has which features? Okay. Now vector carry the DNA from one place to another place. Vector is nothing but which one is used as a vector? Bacteriophage used as a vector, cosmid used as a vector. Okay, and then plasmid and cosmid both are different. Okay, plasmid and cosmid both are different. So plasmid, cosmid, and then bacteriophage is used as a vector. Okay, now we discuss about a vector. Vector. Okay, what is a vector? Carry the foreign DNA from or carry the DNA from one cell to another cell. It's called what? Vector. So which one is used as a vector? Plasmid, plasmid, and then bacteriophage. And then one more enzyme is there, ligase enzyme. Ligase enzyme joins the water, alien DNA to the vector, or DNA to DNA also it's joined. DNA to DNA also it's joined. Okay. Now vector. Vector. What is the vector? It's carry the gene. It's carry the gene from one cell to another cell. From one cell to another cell. One cell to another cell. It's called vector. Okay. Then now vector diagram. Now vector diagram. Okay. Vector, which vector? Plasmid PBR. PBR 322. That plasmid number. B for Bolivar, R for Roderick, P for plasmid. Okay, P for what? Plasmid, B for Bolivar, R for Roderick, scientist name. Okay, then these are what the number of plasmid. Then now and just today I explained the water plasmid description, and tomorrow we discuss about plasmid features. Okay. Now plasmid is also double stranded extra chromosomal. These are also double strand. These are one strand, second strand. Okay. These are double stranded extra chromosomal circular DNA. Circular DNA. Okay. Now in this region have what a ori region. In this region have what a ori. Ori means what? A origin of replication. Ori region means what? A origin of replication. Okay. In this region have what? A ori region. ORI. ORI means origin of replication. Then from here to here empty space. Then from this region to this region. From this region to this region have what? Which gene is present here? Ampicillin resistant gene. Ampicillin resistant gene is present. In short form, it's called AMPR. A for ampicillin. AMP means ampicillin. R for resistance. Okay. These are code to the sites, restriction sites. PST1 and PVU1. These are code to the what? PST1 and PVU1. Okay. From this region to this region. From this region to this region has what? Eco R1, CLAV1 and HIND 3 region. Okay. E. coli, eco restriction enzyme 1. Then CLAV1, CLA, CLAV1. And then HIND 3 region. HIND 3. Okay. Then this site. Opposite to the ampicillin site, here has a what? Tetracycline resistant gene. Tetracycline resistant gene. So TET, R for resistance. These are code. Means from this region to this region have what? Tetracycline. And these are code to the what? Sol and BAM H1. First one is BAM H1 and Sol1. BAM H1 and then Sol1. These are all our restriction sites outside the plasmid. These are all our restriction sites. Then now here rope region is there. Here rope region. At the rope region generally protein is cultivated. Okay. Rope, ROP region. ROP, rope region. These are code to the what? PVU2. These are code to the what? PVU2. Okay. These are which plasmid? These are which plasmid? Plasmid. P for plasmid, B for Bolivar, R for Roderick. Their number is 322. Okay. Now, just now we discussed about that features of plasmid. Okay. First one, it has origin of replication. Second one is selectable marker. Third one, cloning sites. Okay. Third one is cloning site. Now we discuss this one. And first one is origin of replication. Origin of replication. Second one is what? Selectable markers. Second one is selectable markers. Third one is what? Cloning sites. Now we discussed one by one. Cloning sites. Okay. And then say origin of replication. When alien DNA or foreign DNA ligate at the which region? Uri region. Okay. Alien DNA, because of this Uri region have what? Replication capacity or replication ability is there. This Uri region have what? Replication ability. This replication ability not present at this region, not present this region, not present this region. Only origin of replication has what? Which capacity? What a replication capacity. That's why that foreign DNA or alien DNA ligate at the Uri region. Okay. So this Uri region decide the what? Number of copies, number of fragments, decide DNA fragments. For example, number of more than one Uri region are there, then join to the what? More than one DNA fragment. These are DNA fragments. First one, origin of replication. Okay, alien DNA or foreign DNA ligate at which region? Uri region. Because of what? Because of this region. Because of this region has a what? Replication ability. Because of this region has replication ability. Okay. Then these are control the copy number. Uri region control the what? Copy number. Control the copy number. Okay. Then next one is selectable marker. So what is the selectable marker? Generally, antibiotic genes are select the transform and select the what? Transform cell and eliminate the untransformed cell. Okay, first, what is the transform cell? A piece of alien DNA introduced into the host cell is called transform. If that alien DNA not introduced in the host cell, it's called what? Non-transfer or untransformed cell. Okay, what is the transform cell? Okay, these are origin of replication. Second one is what? Selectable markers. So what is the function of selectable markers? Selectable markers select the transformed cell and eliminate the what? Untransformed cell. Okay. So we have how many selectable markers in the plasmid? Generally 4 types, but in here only 2 types is given. Okay, ampicillin, then tetracycline, and kanamycin, and then chloramphenicol. These 4 are what? 4 are antibiotic genes. Antibiotic genes. These antibiotic genes are select the what? Transformed antibiotic genes are select the what? Transform cell. Select the transformed cell and eliminate the untransfer or non-transform. Or untransform or non-transform. Okay. Eliminate the what? Untransform or non-transform. What is the transform cell? Transformed cell means transformed cell means a piece of DNA introduced to the host cell. That cell is called transform cell. Okay, a piece of DNA introduced into the host cell. Okay, host cell is called what? A transform cell. It's called what? A transformed cell. Is called transform cell. Okay, these transform cells are selected by the what? This selectable markers. Okay, and eliminate also. Right. The next point is what? Cloning sites. If one cloning site is better or more than one cloning site is better for cloning, few cloning sites or one cloning site is preferable to the cloning, means only one cloning site is preferable than more than one cloning site. Okay, one cloning site is preferable than more than one cloning site, because of if more than one cloning site is there, okay, we clone the what? More than one gene. These are complicated to the cloning. These are what? Complicated to the cloning. So one cloning site is preferable more than one cloning site. Okay, these are what? Cloning sites. Okay, then tomorrow we discuss about these are not completed totally, and tomorrow we discuss about the cloning sites. Okay.